Okay, my name is Bock, and I'm a second-year PhD student in biological sciences, co-advised by Dr. Kitty Gehring and Dr. Nancy Johnson. I study fungal networks, which have gained a lot of attention in the last few years. In the upper left picture, you can see what we currently know about fungal networks. We know that a certain type of fungus called mycorrhizal fungi can create connections among plants and help transfer resources like nitrogen, phosphorus, and water among those plants. These resources are vital to plant health, and plants trade the products of photosynthesis with these fungi in order to get more of those soil resources that fungi have an easier time obtaining than plants. Our knowledge about mycorrhizal networks has already had huge implications for understanding forest ecology, conservation, and agriculture. These findings have been so compelling that fertilizer companies have even started including mycorrhizal fungi in their commercial fertilizers to be incorporated into farming and even your personal potting mixes. This is what you see in the picture on the left, and my research is testing whether what is actually happening is more like the picture on the right. While we know a lot about that first type of fungus that I talked about, mycorrhizal fungi, scientists have not assessed whether other types of fungi are also creating fungal networks and doing the same sorts of behaviors as mycorrhizal fungi. Additionally, it is possible that other types of fungi are part of the fungal networks occurring and add to the effects of these networks. I'm studying a non-mycorrhizal type of fungus called dark septate endophytes, which are present in many ecosystems and interact with many types of plants. My hypothesis is that dark septate endophytes also create networks and help connect plants in webs more massive than we had previously thought. I began testing this hypothesis in the lab where I grew 2 plants that were only connected by a dark septate endophyte. I created special growth chambers where 2 plants would be separated by a mesh that is small enough only for this fungus to cross. Because of this setup, any nutrients that are added to one chamber and show up in the other chamber could only have gotten there by transportation from the fungus. In my experiment, I added dyed water to one plant to test if the fungus would bring that water to the other plant, which would show up with the dye. If you take your attention to the picture in the lower left, you can see my results. These are microscope images where on the left, there's a plant root with no fungus and no dye. On the right is an image of the plant root the fungus brought the dyed water to in my experiment, which means that the fungus did actually bring water from one plant to another. This is the first evidence in this field that non-mycorrhizal fungi may behave in network-like ways, and my next steps are to use stable isotopes in field experiments to further test my hypothesis. If supported, this would have implications for how we understand what is possible for plant-fungal interactions and how these relationships can be leveraged to improve plant health in conservation and agriculture. Thanks.